Hi everyone, this is Annie, and I'm going to be showing you the method of how to complete a differential scanning fluorimetry or DSF melt curve. So this video is going to cover some background, the methods of the experiment, and then finally where to find the stuff in our lab. So differential scanning fluorimetry or DSF melt curves are used to determine the melting temperature of a protein or a protein-ligand complex. And the way it works is you combine a protein or protein-ligand complex with a dye that when the dye binds to a hydrophobic region, it fluoresces. And so as you increase temperature and your protein unfolds, more and more of the internal hydrophobic regions are exposed, the fluorescent dye binds and you measure the resulting fluorescence. It results in a graph that looks like this, where you start at low fluorescence, increasing the temperature, moving to the right, and the fluorescence increases as the protein melts. The melting point is determined at the inflection point, and so that's about when 50% of the protein is unfolded, but 50% typically means non-functional, and so that is counted as the melting point. So now, how to actually do a melt curve. First of all, safety first. Always wear the appropriate PPE. So in this case, that would be a lab coat and gloves. Now we first prepare our samples. So first thaw your proteins and ligands on some ice. You then dilute the provided DSF dye solution from 1000x to 8x with water. Then you can make mixtures of your ligand and protein samples with DDH2O. And a pro tip is to prepare 4 replicates, so using 5x well volume, so you have always extra volume, for each sample to ensure that you have statistically significant results. You want to run 4 of each sample that you have. Next, you mix your reactions in a microamp fast optical 96 well plate with a barcode. You place the 96 well plate on ice. I always make a 96 well plate map, noting which samples go in each well, and I keep that next to me while I work so that I don't get confused as to what should go where. Then you add 5 microliters of the protein thermal shift buffer to each well. You then add 12.5 microliters of your protein ligand mixtures to each well. And then you finally add 2.5 microliters of your 8x dye dilution to each well. Step 3 is applying the optical adhesive cover to this 96 well plate. You retrieve the adhesive cover. It looks like this, kind of like a sticker. You peel the large middle panel off. Careful not to touch it. You don't want fingerprints on this. Using the edge tabs, which you can still hold, they aren't sticky, you place the adhesive cover on the plate. Then, using firm uniform pressure, you want to press the cover onto the 96 well plate to seal the wells. I use my ID covered in parafilm and spray with 9 or 70% ethanol, but that's just what I suggest. It gives a good uniform pressure across the whole plate. Then you can remove the edge tabs that you're using to handle the adhesive cover. The 4th step is centrifuging the plate at 1000 RPM for 1 minute at room temperature. You place the 96 well plate into a plate basket. It looks like this. Then you centrifuge at the given parameters, and then you return the 96 well plate to ice. So some pro tips on this one. You always want to use a buddy 96 well plate to balance the centrifuge. You never want to run an unbalanced centrifuge. I usually use just an empty 96 well plate, and I mark which one is which. And then finally, you want to keep the plate on ice after you centrifuge it until you're ready to load into the qPCR. You want to keep all those samples cold. Now, step 5 is setting up the qPCR machine for your melt curve. The first step is opening Quant Studio Design and Analysis Software version 1.4.1 on the computer associated with the qPCR machine. I find it just by searching it in the Windows. Next, you want to start your new experiment, editing the following on the first page. You need to add your initials, or I add my first initial, my last name, the barcode from the 96 well plate, the block type, in this case, it's a 96 wells 0.1 mil block, your experiment type, a melt curve, and then for chemistry, you want to select other. Next, on the next page of the setup, you define the thermal profile as shown. So pro tip is to click the minus sign at the bottom of step 2 to remove it. It automatically has a step 2. You don't want that. And the rest you click on the value and then type in to edit to make your changes. It should look like on the right when you're finished. On that same page, you're going to click action and then optical settings. It's at the top right. And then check the box for the X1 470 plus or minus 15 and M3 586 plus or minus 10 option. All the other boxes should be unchecked. So I showed which box that is. It's in the lower table, middle row. Every other box should be not checked. Then you'll need to set up the actual experiment you're running. So you're going to label, assign target, and comment the contents for each well. You can click and drag or click and control or shift click to label multiple cells at once. This is useful since you're doing 4 replicates of each sample, and each sample should get its own target. That'll make sense while you're doing it. I usually call them like target A, target B. In the advanced setup on the same page, you need to assign the tasks. You're going to do U for all samples. And then you set the reporter for ROX for all wells with samples. An example of that setup is shown on the right. Finally, you load the 96 well plate into the qPCR machine, and then you hit start run in the top right corner of the software. And your DSF is running. Be sure in this time to clean up your bench and take care of all your samples, snap drying or snap freezing things if you need. And then another pro tip is that the dye dilutions that you made is biohazardous waste. And that's how you dispose of that. All of the samples that you mixed are biohazardous waste. Obviously, you're using proteins and things. So you'll dispose of your 96 well plate when you're done as biohazard waste as well. So once your melt curve is complete, you need to analyze the data to determine the melting temperature. Using a flash drive, you're going to export an Excel of all the experimental data. You're also going to want to save the images of the melt curves onto that flash drive. There'll be a melt curve plot with the fluorescence and then another plot with the first derivative. Then you determine the melting point as the inflection point or where the first derivative reaches a minimum. So that's shown on the right here. The way that the software outputs the melting temperature, it tends to give multiple melting points just because of the noise in the data. It finds various inflection points just due to the squiggles. But I've found that finding the minimum of the first derivative as the simplest and quickest method of actually pinpointing the melting temperature. And so that is what I suggest you do. So congrats, you've completed a DSF melt curve. And now, where to find the materials in the lab, because I know that can be a pain. So looking south, standing by the sink, this middle picture is our lab. And if you go to the left, the second shelf over Shananda's bench, you'll find the microamp fast optical 96 well plates with barcodes. It's behind a few other boxes since it's used less frequently, but it is up there, I promise. Then going to the right, you'll find above Annie's bench, my bench, on the first shelf, the adhesive optical covers and the 1000X dye. Then the thermal shift starter reagent kit includes a control protein and ligand if you want to run a control to make sure that your temperatures make sense. Those are stored in the -20 on the top shelf. Then the plate baskets for centrifuging are in the Huang lab culture room. So IQ 4539 under their large centrifuge in this box that I've marked with a circle. I do use their baskets, but I do not use their centrifuge, especially because this is a cell culture room. I want to respect the cleanliness of that space. So I take out the baskets back to my bench, thoroughly clean them before and after use, and I use the communal centrifuges. And finally, the star of the show, the qPCR machine is in the middle hallway, directly across from our 4 degrees refrigerator. I'm sure you've seen it, but I don't know if you've ever used it. So that's where it is. A more detailed protocol can be found in the Wildering Lab Share Protocols folder. It's still in the works. I made it while I was running some of these melt curves. So it's draft, but it definitely has a lot of detail for you. And the ThermoFisher user guide is also super helpful for troubleshooting and giving some specific tips and tricks. Thanks so much.